Hi, and welcome back to my channel, Learning Biology with Dr. Vanessa. In today's video, we are going to take a look at the innate versus adaptive immunity. This is part 2 of a two-part series. If you missed out on my last video, which is part 1, please stop and take a look at that video first. That video predominantly goes over innate immunity. In today's video, we are going to take a look at adaptive immunity, and at the end, we are going to close with a comparison between innate and adaptive. In the last video, we talked about innate immunity and how it's a nonspecific, very rapid response, so it comes into play first. But what if that innate immunity is not enough to get rid of whatever may have come into the body? This is when the adaptive immunity takes place. Adaptive immunity is a very specific immunity. It's also referred to as acquired or programmed immunity. Now, this immunity is specific because the response that happens is aimed at a particular non-self antigen. Now, an antigen is a substance that is recognized as foreign by the body, and it provokes an immune response. This means that the adaptive immune system is capable of recognizing the difference between self and non-self proteins. The adaptive immune system does this through what is called basic training. This is where the cells learn to recognize self-antigens and to avoid attacking them. This is referred to as self-tolerance. If this skill is not mastered, then those cells are destroyed. However, sometimes those cells can bypass this, and if there is a loss of self-tolerance, in other words, the cells now recognize self-proteins, then that results in an autoimmune disease. I am not going to focus on autoimmune diseases in this video, but just so that you know, obviously this sometimes does get bypassed, and if these cells do recognize self-proteins, that does result in an autoimmune disease. However, these cells do go through basic training so that they only recognize non-self proteins, because we only want these immune cells to recognize non-self proteins and have an immune response against those foreign invaders that may come into the body. The adaptive immune system involves two types of lymphocytes. They are B lymphocytes, also known as B cells, and T lymphocytes, also known as T cells. Before we take a look at what these lymphocytes do, let's take a look at where they come from. Lymphocytes are a type of white blood cell. Now, both red and white blood cells actually originate from stem cells that are found in the red bone marrow. What we can see here on top is the stem cell, the multipotential hematopoietic stem cell, from which all red and white blood cells are going to differentiate. This stem cell can then differentiate into either a common myeloid progenitor or a common lymphoid progenitor. Now, the myeloid progenitor, real quick, gives rise to red blood cells and then eventually thrombocytes, which are involved in blood clotting. It also gives rise to things like mast cells and the myeloblast, which makes basophils, neutrophils, eosinophils, monocyte. These are involved in the innate immunity that we talked about last time. And then if we look over here, this is what we're interested in this time with the common lymphoid progenitor. You even see it in the name. These are where the lymphocytes come from. So we have natural killer cells. Remember, natural killer cells were talked about in the last video. These are actually part of the innate immune system. And then we have the lymphocytes that are going to come from the small lymphocyte, differentiated into T cells and B cells. Now, T cells actually will leave the red bone marrow and they will mature in the thymus, but the B cells are actually going to complete their development within the red bone marrow. Because there are two types of cells, main cells, that we see in adaptive immunity, the B cells and the T cells, there are actually two types of adaptive immunity that come from these cells. One of them is referred to as cell-mediated immunity, and the other one is referred to as antibody-mediated immunity. In cell-mediated immunity, cytotoxic T cells will work by directly attacking invading antigens. They do so by attacking cells that display portions of this antigen on their cell surface. In antibody-mediated immunity, this is the work of B cells, and B cells will actually recognize antigens, and when they do so, they become activated, and then these B cells turn into what is called plasma cells. When a B cell is activated and turns into a plasma cell, it will then start to secrete antibodies against those specific antigens. Now, both of these processes are very specific and can be complicated, but they do also use the help of another T cell that is referred to as a helper T cell. So a helper T cell can actually cross over and work in cell-mediated immunity, and it can also help in antibody-mediated immunity as well. The important thing to note here is that in both of these processes, the recognition of the antigen is specific. So whether we're talking about cell-mediated immunity or antibody-mediated immunity, we're talking about a specific binding that is going to occur, a specific recognition. This sets adaptive immunity apart from innate immunity. The other thing is that when this process occurs, the other thing that's made is memory cells. And the memory cells can either be memory T cells or memory B cells, depending on which process we're talking about. Memory, again, is something that is specific to adaptive immunity. This is also interesting because now it takes a while for this whole process to occur initially. But the second time that the body comes into contact with this foreign antigen, it can be much faster because of this memory, because of all these memory cells that are made. The second response can be much quicker. In order to better understand how these cells work, we need to first talk about the MHC, or major histocompatibility complex. These MHCs are self-specific. That means they are very specific to each individual person, and they are actually alien to every other person. Their task is to actually bind self and non-self antigens, and then to display those antigens on the cell surface. Now, there are two types of MHCs. We have class 1, which is referred to as MHC1, and class 2, which is referred to as MHC2. MHC1 is found on the membrane of every body cell except red blood cells because they do not have a nucleus. MHC1 show antigens that are internally produced. That means what is shown on the surface of these cells are things, proteins, etc., that were found inside the cell to begin with. These internally produced antigens are referred to as endogenous antigens. And so because they can display both self and non-self antigens, this cell can be healthy. They will always show different things on there. Basically, what this is the cell is showing the immune system what's going on inside of that cell, so then the immune system can determine if whatever's going inside is normal or should we mount an immune response. So in a healthy cell, the MHC1s will have normal body proteins inside of it, and when an immune cell comes by, it won't mount a reaction because in a normal response, the immune cells should have been conditioned to not recognize self proteins. And so if the cell is happy and healthy and normal and showing normal proteins on its surface, the immune cell will just keep wandering by and not have a response. However, let's say, for example, that specific cell got infected with a virus. Now it's going to be displaying non-self proteins on its surface. And when the immune system comes by, it's going to recognize that those proteins in the MHC are foreign, and it's going to mount an immune response against that cell. So in turn, this is basically the cell's way of being able to tell the immune system either I'm healthy, I'm fine, everything is normal, or there's something wrong inside of me, destroy me now. Now, the other type of MHC that we see is a class 2 MHC, which is also referred to as MHC2. These MHCs are only found on certain immune cells that are capable of phagocytosis. So these cells are capable of eating things. So these are going to be things that are found outside of the cell. Now these cells, because they are also body cells, will also display MHC1. So these specific immune cells that are capable of phagocytosis can actually display MHC1 and MHC2. But the MHC2 is only found on these cells that can undergo phagocytosis. And so these are going to be exogenous antigens, right? Antigens that were found outside of the cell and taken in. So they take it in, and as I had mentioned before, they break them down, and then some of those proteins can be displayed on the cell surface. These cells are also referred to as antigen-presenting cells because of this capability. And so if the antigens that they're showing on their cell surface are normal, again, the immune system can just pass on by. But if it happened to eat something that was abnormal, not a self-protein, then the immune system can mount an immune response. Now, let's briefly talk about cell-mediated immunity and how a cytotoxic T cell is triggered to be activated. This is actually really complicated, so I'm going to go through a very basic overview of how this works. So first we'll start with a cell. And remember, this can be a body cell or an antigen-presenting cell as long as it is displaying an MHC1. Now, if this MHC1 is displaying a non-self protein, then a cytotoxic T cell can recognize that that MHC1 antigen complex is not self. And it will attach to that with a receptor that matches to the receptor that's on the body cell or, again, the antigen-presenting cell. Once this happens, the cytotoxic T cell is now activated and divides to produce many clones. Some of those clones are going to include memory cells. So the clones will be activated cytotoxic T cells, which are going to be present in this very immune response, and then memory cells, which will be able to be present in a subsequent immune response. The activated cytotoxic T cells can then go and recognize other cells, other body cells that have this MHC1 antigen complex on it, and then it can bind to those, and now because it's activated, it can actually release substances that induce apoptosis of the infected cell. So once it's activated and then it goes and attaches to MHC1 on body cells that are displaying this specific antigen, it's going to release granzyme and perforin, which actually perforate the cell wall of the infected cell, and it's going to cause that cell to undergo apoptosis, which is cell death. Now, in antibody-mediated immunity, we are going to be talking about the activation of B cells. I've already made a video animation. It was actually a collaboration with a student that shows this B cell activation. Now, B cells can be activated in two ways. Either they can be activated by a free-floating antigen within the blood itself, or it can be activated by a T helper cell. I'm going to go through both ways that this happens. First, we're going to talk about B cells that are activated by the free-floating antigens. Now, B cells have B cell receptors on their cell surface, and this is really unique because every B cell has a lot of these B cell receptors around the entire cell. I'm not going to draw all of them, just drew one so that you can kind of see that idea. But they have a lot of these, but they all recognize the same thing on that one B cell. However, there's a lot of variability, so each B cell differs from another B cell. And the idea behind this is that B cells can basically react to anything that they may encounter that is not a self-antigen. So what's going to happen is if this B cell comes into contact with a free-floating antigen, so there's some antigens in the body, and it comes into contact and it binds, then that is going to be able to activate that B cell. That B cell will actually internalize this receptor because that receptor specifically bound to the antigen. It's going to internalize it, and then the B cell is going to go through clonal expansion. When it goes through clonal expansion, it's going to make memory B cells. Again, remember that's very specific to this adaptive immunity. It's going to make memory cells. These are going to be cells that are around for the next infection. And these memory B cells will be identical to that activated B cell. So same receptor that was found to bind to that antigen would be the same receptor on those memory B cells. Also will be made are activated B cells. And activated B cells are referred to as plasma cells. Now remember, I just said that the B cell will internalize that initial receptor, and these activated B cells or plasma cells are going to start pumping out antibodies that are identical to that receptor. Now, why is it important that it's making the same exact type of antibody as the receptor that was internalized? Now, it's important because they're going to recognize whatever's in the body. It's the one that gave the initial response. It's the one that's going to be made. So antibodies will be made in massive quantities from these cells that are activated. Now, on the B cell, the receptor on the B cell is actually IgD, immunoglobulin D. And there are 5 different types of antibodies. When plasma cells are activated, they will actually make IgM first, and then they will class switch into IgG eventually. I have another video on antigens and antibodies where I talk more about antibodies in general. So once this happens and these B cells are now activated into plasma cells, they're going to start making those antibodies, and now those antibodies can go on to do their work. And antibodies can do things like bind to the antigen and neutralize it so that that antigen cannot do any harm. Now, the antibody itself cannot destroy anything, but it can bind to that antigen, preventing that antigen from doing what it was supposed to do. It can also work in opsonization, which means that it can flag that antigen and then other immune cells will come and destroy it, eat it up, etc. What about MHC2? I mentioned MHC2 before, but we haven't really talked about it. MHC2 is responsible for activating T helper cells. If an antigen presenting cell displays a pathogen that it has internalized, remember MHC2 are found on antigen presenting cells. If it presents this pathogen that it has internalized, then it can activate a T helper cell that recognizes it with its T cell receptor. If the T helper cell recognizes this MHC2 antigen complex, then it will go on into clonal expansion, and memory helper T cells will also be made, and then activated helper T cells. When the helper T cell is activated, it can then go and bind to a B cell and activate a B cell. Now, this is interesting because B cells are actually also capable of phagocytosis, and so B cells can also display MHC2 on their cell surface. This gets really complicated of a process, but T helper cells can also help to activate B cells as well. So B cells can be activated by that free-floating antigen I just talked about before. They can also be activated by an activated T helper cell. So if an activated T helper cell binds to a B cell, it can activate that B cell, and then that B cell will go through clonal expansion, making memory B cells and activated B cells, which are plasma cells, make an antibody like I just spoke about. Helper T cells can also help to activate cytotoxic T cells. So now that you've looked at the difference between the innate immunity and adaptive immunity, let's put it all together and look at the differences and also similarities between the two, like some cells that may be used in both. So first of all, overall, the innate immunity is nonspecific. It is a rapid response, while the adaptive immunity is specific. It is a slower response the first time, the second time it is quicker, and there are memory cells involved. Let's break it down into the types of cells we will find in each. In innate immunity, we see mast cells, neutrophils, and monocytes. In innate immunity, we also see dendritic cells and macrophages, which are antigen-presenting cells, and natural killer cells, but these will go in the middle because they actually overlap some of their responsibility in engaging the adaptive immune response. And finally, in adaptive immunity, we see B cells, and T cells can be cytotoxic T cells and helper T cells. I hope that this two-part series helped you to clear up the difference between innate and adaptive immunity. Let me know in the comments below, did you like this? Did you like this series split up into two, talking about innate immunity in one and adaptive in the other, and how I brought it all together in the end? Do you have any questions or comments? Make sure you drop those down below as well. As always, please make sure to subscribe, like my videos, share them with your friends. In order for my channel to keep on growing, I appreciate the support, and as well as questions and comments on this video, drop down any future videos that you may want to see. Thank you.